Okay, all right. So as I was trying to say, we are working with a specific animal model in my lab, which is called the non-human primate. The non-human primate, or known also as the monkey, is right there in the middle at a very sweet spot between two other animals that we use in research a lot, like the mouse, the rodents, and the rats, and then the people that we're trying to treat, which is us, the human patients. And we're using the non-human primate because it is very similar in neuroanatomy, in genetics. The macaque monkey, for example, shares 95% of their genetic codes with humans. The size of the brain is also at a sweet spot, meaning that if you're trying to develop something that will have real world applications in humans, the monkey is like the perfect jumping board, okay, to get there. Although it is a small part of what is being done in biomedical research, monkeys only represent 0.5% of all animal in research. They represent 70% of all new treatments that make it to clinical trials and are changing people's lives today. That includes, for example, L-DOPA that my grandfather took when he developed Parkinson's disease, deep brain stimulation, where people are implanted with stimulated electrode in their deep brain to alleviate some of the symptoms, tremors, and then some of the most recent stuff like brain-machine interface. Maybe you guys are familiar with that. Here, this is one of the first patients that's already whole from BlackRock Microsystem. This is a paralyzed patient that has an implant in their brains, in the motor cortex, the part of their brain that's controlling movement of the arm. She's thinking about moving her arm. Of course, her arm is paralyzed, but then it's moving this robotic arm, okay? And of course, where did that came from? Well, that came from tests in the monkeys. Right here, as you can see, the monkey is feeding himself marshmallows using only the power of his thoughts. And even more recently, you guys have probably seen that Neuralink has implanted their first patient. And this patient is implanted with kind of a more fancy version, a wireless version of these brain implants. And this patient, as he's having a conversation with the interviewer, is able to play chess on a computer. He plays Civilization. You've probably seen this video. This kind of went around the world. And again, where did that come from originally? Here at the bottom right, traces of multiple neurons. And maybe you remember this video that came a year before, the Pong video, where the monkey is playing a video game there where he has to move the joystick to hit a certain box. But as you can see, the joystick is actually unplugged. And the monkey is not using the joystick. He's thinking he is. And the fact that he's thinking about moving the joystick, it is these thoughts that are actually moving the dot on the screen through the neural link, through the brain implant. So these are the kind of things that fascinate me. We've also used monkeys to understand the neural computations of cognition for a very long time and made some major breakthroughs. For example, decision-making in the Shadlin lab at Columbia, where the neural correlates of decision-making have been very well characterized. Working memory, this is a more recent paper in Science showing how we can use geometry of neural subspaces to understand how memory is stored in the brain, and then movement, like the Mark Churchin lab. Mark Churchin, big figure in the computational neuroscience world that you probably know. This type of computational work is the foundation for some of the brain-machine interface that you saw before. But where does that all start, okay? The signal that we are recording is the most beautiful signal that I've ever heard. I remember when I did my PhD at McGill, and for the first time I entered a room where there was a monkey and somebody was recording, and then he had a headset on, and I was thinking, what are you listening to? And then he's like, listen, and then he turns the volume on the speaker on, and you could hear this neuron. Very, very clear signal that is the action potentials of these neurons over time. And this is the neural code that we as neuroscientists need to decode. We need to understand that. This is the communication. It's the information currency in our brains. So this is a signal that I recorded in the lab where I did a postdoc at the MNI. And that's another thing that I want you to appreciate, just the beautiful waveform of a single neuron being recorded. You have an electrode that goes close by a neuron, and extracellularly, you're able to capture these voltage fluctuations. And look at the signal to noise ratio, right? Like this is noise, this is signal, right? Like we're not, we don't even need statistics to understand this. We need barely any pre-processing. This is real information. It's as clean as it's going to get. And that's what really made me fall in love after many years of doing neuroimaging in humans, made me fall in love with neurophysiology. And what's the cool thing is that you can do that while the monkeys are performing tasks here. This monkey is performing a specific conditional task that I'll show you a little bit later. But the source of some of the understanding of the neural code came from studies of the visual system. And you've heard about Yule and Weasel, for example. And while I'm talking, actually, I just figured I brought here a 3D printed monkey brain. This is Coco. It's actually one of my monkey. And we do MRIs of these animals and then 3D printer brains for neurosurgical preparation. So I'll just pass that around on one side. And just to give you a sense of the size here, this is a skull of a macaque monkey. You can be careful with the canines here. You can just pass that around. Here, going back to understanding the neural coding of visual system, the very early work here, essentially what you do is you listen to the number of action potentials that a neuron is firing. It could be slow or at 100 times a second. And then what you realize here in this experiment, Weevil and Weasel presenting bars of different orientation, listening to what the neuron would react in certain orientation. The neuron didn't react at all. And then a specific sweet spot orientation, then the neuron would go crazy. And essentially you do that for all possible orientation, and then you end up with something like this, which is the firing rate of the neuron as a function of the orientation of the bar, and then there's a sweet spot, and this is called a tuning curve, right? And I'm like, oh wow, maybe this is how information is coded by a neuron. They all have like these tuning properties. So let me show you now outside of the visual system when we're trying to understand something a little bit more complex, like cognition. So here is a task that you can train monkeys to do. You can train monkeys to do some amazing stuff. I trained monkeys to play Unreal Tournament. First-person shooters. We've trained monkeys to play on the stock market, to play Battleship. They're smart animals. You think your dog is smart, your monkey is a lot smarter. So to start a trial, monkey presses on the touchscreen, sees a stimulus here, a red flower, then there's a delay of a few milliseconds to a second, and then two target appears, and he makes to make a decision. If you've seen this, then he needs to select a star. So he needs to learn these rules, whereby if first you see a red flower, select a star. A white flower, also select a star, or a yellow flower, then you need to select a circle. These things appear all over the place. They're randomized in location. And as we're doing that, of course, we're recording the neural activity. The monkey learned around 40 of these associations that are completely arbitrary. This is called conditional associative learning. It's the kind of thing that you use when you're driving your car and you see a red light and you know, oh, I need to stop my car. A green light, I need to go forward. There's nothing intrinsic about red or green that tells you forward or stop. These are conditional associations that we have to learn as we grow up. Monkeys learn that. So let me give you like a kind of a very simple single neuron example of how a neuron would code something as complex as a conditional association. So here, this is plots that are called raster plots. Every little dot that you see here are action potentials for a neuron, and these are repeated trials. So we repeat multiple times. The monkey does many times, many trials. And some trials, the target is actually the circle, and other trials, the target is the star. And so first there's a pre-trial, then there's the cue, like a red flower, for example, and then there's a delay epoch of varying length. And just by looking at this plot here, you can see a massive difference in the response of this neuron during the delay epoch when there's nothing on the screen, where the monkey doesn't even know what he'll need to move, what movement he'll need to plan first. You can see that the neuron will go crazy if the monkey knows he will have to select a circle eventually. So this is not a visual correlate. This is not a motor correlate. This is a cognitive correlate, neural correlate. So here, this is raster plot, again, multiple trials. Now, what people do, neurophysiologists, often when they do single neuron analysis, they average out the noise. And I want to talk a little bit about what noise means here. So noise in the mind of a neurophysiologist means every time you present the same stimulus to a neuron, to a monkey / neuron, the response of the neuron is not exactly the same. And it should, you know, ideally present the banana, 10 spikes, present the banana, 10 spikes. No, it's more like present the banana, 8 spikes, then 12 spikes, then 13 spikes, then 9 spikes. like there's variability from trial to trial. So people, to account for this variability, they average across multiple trials, and then you get something called a spike density function, okay, which is essentially just a smoothed curve of this activity. And here what you can see is in the case where he needs to select the blue circle, then this neuron will fire very high during the delay epoch. And if for the green star, it'll fire below baseline. And you can see that this neuron doesn't care which of the two cues were represented. They only care about what is the meaning of that cue, right? The responses are indistinguishable based on what cue is being presented between two cues that point towards the same target. So this is just to give you a little example of what single neuron information can give us. But the world has evolved beyond single electrode recordings. And this is where finally I'm going to get into AI and machine learning. So for example, an implant called a Utah array really revolutionized neural recordings about 15 years ago, okay? And this is something that was been developed by BlackRock Microsystem, used to be Cyberkinetics. And instead of recording from one single electrode, and you record from 100 electrodes that are implanted chronically inside the brain. And these arrays are pretty expensive. They're around $10,000. I have one right here. This is my practice one. So I allow you to open the box, but not to touch the array, okay? But here you will see in this array, the connector part that I'll show you just a little bit later. That's called a center port. And then the array here. And you see also some reference wires. So you can pass that around. So that makes it a little bit more real for you. This was already implanted in 2 brains. So it's been cleaned now. All right. So now going to multi-electrode recordings, what do we get? Well, all right. So we need to implant these Utah arrays into brains. This requires some special neurosurgical techniques. That's one of my expertise, neurosurgery and primates. This is me implanting one of those arrays using a pneumatic pump. And this is the array itself into the brain of a monkey. And then the Ceroport connector that you'll see here just protrudes out of the skin. It's very clean. And here's a patient who has 3 of them in their brain. This is a paralyzed patient. If you go to SFN this year, he'll probably be at the BlackRock booth. You can talk to him, touch the thing on his head, ask him, how does it feel? He'll tell you, it doesn't hurt at all. It's part of my body now. And he will use that to control, just by the force of thought, his wheelchair or a computer. This is someone, Buzz, who had a surf accident not too long ago. So here, when you record from a Utah array, then what you get is a signal that looks like this. Now you have 100 of these channels that are electrodes. And in each of these channels, using spike sorting, you'll detect anywhere from 1 to 5 neurons. So you end up with a data set that's very rich and multidimensional. You're having the firing rates of hundreds of neurons simultaneously recorded while the animal is performing a cognitive task. And when I started my PhD, I was coming from psychology, okay? I hated mathematics. I was like, I suck at this. I don't want to do any of that. And then I realized, all right, there's no way around it. I got to learn some stuff. And where do you start? And for you in the room who feel a little bit lost when you hear about information theory or decoding and so on, this is the paper that got me into it. So you can take a picture of that if you're starting, read that paper. It was just the most beautiful explanation of how to analyze a multidimensional data set like this. So that's a recommendation that I have for you. But let me give you just the gist of that, right? So when you record from a neuron, okay, just let's say neuron 1, then there's the onset of a stimulus. And then, for example, if neuron 1 is a spider, doesn't care about the spider, doesn't fire at all. Each row, again, is a trial. And then if you present the tau pisa, then the neurons fire. And then let's say that's just one neuron. That would be pretty straightforward to plot this. Now you have 2 neurons recorded simultaneously. And let's say the second neuron cares about the spider a lot more than that. So now what you can do is you can plot the activity of these 2 neurons recorded simultaneously on a 2-axis plot, right? Where every dot represents the simultaneous activity of neuron 1 and neuron 2. For example, that dot here means that neuron 1 fired high, and neuron 2 didn't care too much, right? And so here you can see some representations that are slightly different. For example, this would be a representation, a 2-dimensional representation of the spider, and here would be a 2-dimensional representation. Here would be a representation of the tower, right? So that's for 2 neurons, all right? Now, what do we do when we have 300 neurons? Okay, well, that's where machine learning comes in, okay? And there's many different algorithms that probably some of you have learned and know how to use, but if you don't, like one of them that I used originally and has been working very well for many years is called support vector machines. And essentially what you do is then instead of plotting in two-dimensional, then you plot in three-dimensional if you have three neurons. But in four neurons, of course, you cannot imagine a four-dimensional plot, but the computer can, and then you go on 5, 6, 7, up to 300 dimensions. And then what you can do is fit, if you had three neurons, for example, fit a plane that separates the simultaneous activity representation of three neurons for one trial, two trials, and then this is for when a spider is being presented, when the tower is being presented, and you fit a plane. And then if there's a new trial that's being presented, then you can plot that plot here unlabeled, and you can guess, the decoder can guess, well, based on the activity pattern I'm seeing right now, I can predict that this monkey is seeing a spider right now, right? So that's kind of the basis of decoding. So you train the algorithm with a training set, testing set, you obtain something called encoding accuracy. I know this is very basics, but again, gear more towards the beginners here. Kernel tricks, I'll skip that. So how did I learn how to implement it on the computer once I had some theoretical basis from that paper and many others? I imagine many of you have already done this class. If you haven't, this, at least at the time that I did it, again, with very little mathematical background, Coursera, the machine learning specialization class from Andrew NG, just excellent stuff for people who really don't have that background. Get you the tools to run an SVM, but not just like click, like run, and then have the results and then put that on a poster and be proud of it. But actually the quality checks, that's what matters. Understanding what's going on behind it, right? And for something like support vector machine, like you can actually understand what's going on, which is not necessarily the case with deep learning applications. So it's not completely a black box there. Anyway, just saying, this is very cool. To be a good neurophysiologist, you need to do more than know how to run a script. You need to understand everything that's behind it, the pros and cons, the failure modes, and so on. All right, so early paper of mine during my PhD, I was like, all right, can I use SVM, for example, to decode the focus of attention of a monkey? And not the focus of visual attention like you know right now, I'm looking at you. When I could, for example, look behind the room right now, but pay attention here, right? If somebody, that's called covert attention, right? That's what you do, yeah, when you don't want other people to see what you're paying attention to. So essentially, I train a monkey to do that, fixate on dot, but pay attention to different parts of the screen so he could detect something that happens very quickly. And this is a trial where he had to pay attention to the top left, top right, bottom right. And this is the simultaneous activity of about 50, 60 neurons reported from an older version of the Utah array. And already when you look at that, you can see that there are some patterns that seem to be different across these four conditions. So then you do the support vector machine or whatever other machine learning algorithm you like. And then you feed that data into it, train, test. And then what you can get is a decoding accuracy of the neural ensemble that is every neuron is a predictor, right? Is a feature in your decoding algorithm. And then you get a decoding accuracy from testing on unlabeled trials. And then you can see here that during the delay epoch of that task, which is the epoch that matters, when I'm trying to decode attention, I can decode with around 75% accuracy where the monkey is paying attention covertly in his environment. And this is interesting because this is a personal information. Nobody else than you know where you're covertly paying attention to. So with this, it was possible to extract this kind of private information from the monkey and then get some cool results. So here is the full ensemble, and every little dot you see here is from each single neuron. If you just use one neuron at a time and run the train test and decoder on one neuron, you can see most neurons contain no information, and then a bunch of neurons contain a medium amount of information, but a lot of them contain redundant information, right? That's why you don't just have this green line going up the roof. Many of these neurons code for similar things. All right. We were very happy about this, my supervisor and I, this back in my time, Miguel, and just funny story here about one caveat about machine learning and AI. And I hope today to present kind of a nuanced view, because there's a lot of hype about these techniques, and some people think that, all right, we've got it solved, and we're not really. Like, there's major limitation to these analysis techniques, and just let me tell you about that story. So essentially, my friend and I, Matt Levitt, was sitting just beside me on my desk. He was also analyzing Utah array data from monkeys doing a working memory task. And as I published this paper in Neuron, he published this paper in PNAS, and the results support, of course, in my case, that the area I was recording from, the lateral prefrontal cortex, is an area that's important for covert attention. And then he was like, the area that I recorded from is important for working memory. And then only at a later conversation, we understood that we actually recorded from exactly the same area in the brain. And like, how can both of these stories be true, right? So I'm finding some neural correlates here. I'm publishing a paper thinking that these neural correlates represent this function. And he's recording other neural correlates and comes to a different theory about the same brain area. All right, there's maybe a possibility that this brain area codes for both of these things and actually does it. But actually, the lesion studies, when you remove this brain area bilaterally in the monkey, show that those animals have no problem with working memory and they have no problem with covert attention. All right. So probably both of these papers are spurious. Okay, they're meaningless. Okay, and I worry a lot about how many meaningless papers are we publishing today. All right, and I'm maybe a little bit of a skeptic, but I think it's probably the majority of what we're doing. So this is correlational research, right? I'm recording neural data and I am observing how the neurons change as a function of stimulus input. And then that's a causal relationship. I know that it's this stimulus that's causing the variation in my neural activity. But how do I know that it is this change in neural activity, these neurons, that are driving the behavior? For example, covert attention or working memory. That I do not know in a causal manner. I know it only in a correlational manner. And there's many ways in a very complex network like the brain that spurious association can fool you. Okay, so that's just my little warning sign. It's not because you decode something somewhere that you should jump to the conclusion that you found a center for X. Okay. All right. So another cool thing that we can do with monkeys is study natural behavior. Their natural behavior is highly complex. They form lifelong bonds, alliances, hang out in families, have complex exploring mechanisms, love. Chimpanzees even conduct genocides on nearby gangs. It's a very rich world, the world of primatology, and I got to experience that a little bit as I traveled to some islands to study natural macaques in the wild. And one of the things that we wanted to do was to analyze the neural correlates of natural behavior. And this is not something that had been done before. So one thing that we had to do was to shrink that full neural recording data set into something small enough that we could put it on the head of a monkey that is freely moving to do wireless recording. And that's what we did here, developing a wireless neural logger system, which essentially all the neural data is saved on a micro SD card. And these micro SD cards now have like 0.5 terabyte of data storage. So here's a macaque monkey with that device on his head. And right now he's asleep, but we can also record this as he's performing natural behaviors, here in this case, grooming, which is a very important filiative behavior where they establish bonds with one another. And so here we managed to record with Utah arrays, and that's the same Utah arrays that are in human patients, the same neurosurgical procedure that we're using, very high standard of care here. Okay, this is not mouse research. Never lost a monkey in any of my experiments. Okay, it's not like mouse, not working. Oh, that's not how it works. We're treating these animals like they are a little child. Okay, all right. So here we are able to study these animals, their brain, their neural activity of hundreds of neurons as they're performing many different natural behaviors, as they are in group, as they are alone with their partner. We studied everything from aggression to having sex to feeding. And so we've been able to, to characterize the neural activity pattern of the entire breadth of natural behavior in a primate species, and that had never been done before. This is something we call the neuroethogram. The ethogram is the inventory of all the behaviors proper to a species. And the neuroethogram, as you can guess, is the neural signatures for all these behaviors. There's many more than them, but you got to fit it on one slide. And here, as you can see, there's many different colors that seem to correspond to the different behaviors. So there's information in all these neural populations that seem to be characteristic of the different behaviors that we have as primates. So let me just see here. There was a point that I wanted to make about training in natural behavior versus tasks. So most of the stuff that people are doing is this, all right? And that's what I started my talk with. We train monkeys to do tasks. They do multiple trials of this task, and then we analyze the neural data. When there are repeated trials, it's pretty easy to then just average out the noise, right? We average trials of the same trial condition, and then we don't care about the noise. Well, when we record from multiple neurons simultaneously, what's good is that we can do single trial decoding with SVM, for example, and then consider that the noise is somehow not average across trials, but across the neural population of neurons. It's like every neuron casts a vote. Some votes don't make sense. Most of them do. And if you look at the aggregate of it, you'll get something that's actually quite meaningful. But what do you do when you analyze natural data, right? I say a single trial, also called life. Like my life, like if I go and I stumble here, I don't get another trial, okay? I really just stumble, all right? Life is a linear timeline, okay? You have one shot at it, okay? So that's what we're interested in, particularly. How do you analyze that? How do you get rid of noise? How do you train a decoding algorithm? You don't have a training set, right? You don't have repeated labels of the same things. Like every moment is unique. And so here, what I'm trying to bring forward is that we didn't know how to analyze this data set, all right? Like we did our best shot at it, and I think that we did okay. But there's definitely a lot more richness to this data set that we have not explored. This was kind of a first trial at it. One thing that we could do, for example, is just a nice visualization here. This is called UMAP. And let me know in time because we kind of started off. So when I have 5 minutes left, please let me know. So here, this is a clustering, unsupervised clustering algorithm. That's one approach that we use. Every dot here represents a second in the life of the individual. And this dot here is represented in a space. That's the space of all the neurons, right? You remember this 2-dimensional to 3-dimensional to 300-dimensional. We dimensionally reduce that to only 3 axes that explain most of the variance. And each dot here is colored by what's the behavior that the monkey was doing at that time, during that second. So you end up with clouds of dots that seem to cluster according to certain behaviors. So that's good. That means that the neural population together has a very specific signature depending on what you're doing. So I want to pay attention. I want you to pay attention to a specific one here where you have 2 clusters. Like why, for example, there's that blue cluster, the blue cluster there, that one. Why are they separated? So here, for example, it's give groom. Give groom, that's I'm grooming my partner. These were couples, lifelong, female and male. Grooming is like giving a massage to your partner. They really like it, just like we do. And so how can this be explained? Well, here, if I recolor the same cloud of dots, not by behavior, but based on the social context here, what we'll see is that if you are grooming your partner and you are sitting beside a male neighbor, or you are sitting beside a female neighbor, the brain registers it, and the neural signature is different. And that makes a lot of sense. When we go to the restaurant, we may be talking to one person, we care about who we're sitting beside, right? This is something that we need to know. Maybe I'm not going to say the same thing if I'm sitting beside a friend or my supervisor, right? So these are the kinds of things that the brain does, how they do it. Maybe this is the way that this is how it does it, right? But this is correlational. I don't know. If I wipe out these neurons in the brain, are you suddenly starting to say crazy stuff in front of your supervisor? We have to run the experiment. All right. So how would you analyze single trial natural neural data set? Okay, I feel that this is the new frontier in cognitive neuroscience as we're moving towards more and more ecologically valid paradigms. Ecologically valid, what it means, more lifelike, right? We've done millions of experiments in highly restrained environments that gave us bogus results that never translated into real life, okay? So here, we're trying to kind of open up a new field, natural or neuroethology, and it's not clear exactly how to do that. And you guys are the future, the people who are interested in data analysis and computational neuroscience. So this is a challenge for you. And if you want a piece of the data set, contact me. If you have some ideas on how to maybe, extract some more juice out of this, you have your own theory, I'm more than happy to collaborate, okay? So in closing, I just want to give a final warning, all right? There's 2 camps. As I've evolved throughout the last 15 years of doing this, I realized that there's really 2 camps that don't talk a lot to each other, all right? There's the experimentalists and there's the theorists, okay? And if you want to know if you are a theorist or an experimentalist, well, if you're spending 100% of your time in front of a computer, you're a theorist, okay? if you're spending any time collecting data, then you are an experimentalist, okay? So I'm an experimentalist. I'm not a theorist, okay? I depend on the intelligence of theorists like you to make sense of my data, okay? And I struggle to record more and more complex data with more channels and more naturalistic paradigms. But when I'm looking at what some theorists are doing, as I talk with my group of experimentalist friends, we feel like it's a little bit like a beauty contest, what you guys are doing, okay? It's like my model is more beautiful than yours. It explains more variance and right? It seems completely unrelated to biology at some point. I'm not saying for every case, but in a lot of cases, you guys have your own circles of theorists and you're optimizing these models. And I think there's a problem to that because I doubt that there is much value to an exercise like this if it cannot be tested in a real world experiment. So I'd like to give you 2 rules if you are willing to analyze this data set or come up with a new theory about how the brain works. Rule number 1, only build models, theories, that generate clear predictions, okay? A prediction means, all right, if all of this thing is true, then there's this one thing that should also be true that I cannot confirm right now in my data set, okay? But that's a prediction, all right? So that's rule number 1, make clear predictions. Second, only build models whose predictions can be tested with actual experiments, all right? So second is I made this prediction, all right, now what is the experiment that could be done to validate this or not? If we don't do that, models do not evolve with data, right? Models kind of evolve in their parallel universe of beauty contest. But we're trying to understand how the brain works here. So we need to hit that gold standard, make predictions, and think of experiments, talk to experimentalists, say, can we run this experiment? And he says, we couldn't run this experiment in 100 years. Drop your model. Do something else. Do something that you can test. Your career is going to be a lot more interesting. You're going to be much more grounded into the real challenges of neuroscience. And you've seen Oppenheimer recently, probably. That's a long movie. But one thing that I liked about this movie is especially that it's the collaboration between theory and experiment. And I remember this scene in the movie where Oppenheimer shows up and then he has to choose this office. And he's like, I want to work in this office. And people are like, why this office? Like, because I want to be beside the experimentalist, right? And that's the other guy, Dr. Lawrence, right? And in this movie and in the reality, what happened is, well, Oppenheimer, was doing these equations, coming up with that. He's like, therefore, this must be true. And then he goes to the next door and he's like, could we run this experiment? He's like, of course. And then he runs this experiment and it's not true. He's like, ah, shit. And then he comes back and then he rethinks it, right? That loop is a meaningful collaboration that I don't see happening in neuroscience. We need that. We really need that if we want to make the same breakthroughs that happen in physics, for example. All right. So I'm starting a new lab here at Université Laval. It's called Translational Neurosurgery Laboratory, but we do much more than just neurosurgery, although it's a big part of what I do. Developing the wireless neurophysiology, optogenetics, and chemogenetics to have causal manipulations of brain. That's very important, and we're doing that in monkeys so that we can get that to clinical trials in humans. I'm located right here at CERVO. If you want to learn more about the lab, here's the email address. If you're interested by anything that I've said today, here's the website, and here's my email address. Yeah, in closing, I think it's a very, very exciting time to be doing neuroscience. You're just like being born in the world at the right time. But we're facing also some important challenges. We need more meaningful collaborations. I hope that I can find among you a community of theoretically minded people who are also open-minded towards experiments, and we can talk to each other. All right. Thank you so much for your attention today.